Hi, welcome to my YouTube channel Bio Explorer. I am Dimut Jayaratna. This is an educational channel and it will be very useful to improve your biological knowledge. Today in this video we are going to talk about cytoskeleton, cilia and flagella, which are few components found in a cell. Cytoskeleton. Cytoskeleton is the supporting structure of the cell and maintains its shape. It is more important for animal cells which lack cell walls. It is a complex network of protein filaments present in the cytoplasm of all cells, including prokaryotes. Additionally, it is dynamic, hence has the ability to break and reform as needed. It extends throughout the cell from the nucleus to the plasma membrane. In eukaryotic cells, there are three types of components in the cytoskeleton as microtubules, actin filaments, they are also called microfilaments, and intermediate filaments. These are all capable of rapid growth or disassembly based on what cell needs. Each type is formed by the polymerization of a protein monomer. Microtubules. They are hollow tubes with a diameter of about 25 nanometers. The wall of a microtubule consists of 13 columns of alpha and beta tubulin monomers. Functions of microtubules: maintenance of cell shape, cell motility. In 9 doublets oriented about 2 additional microtubules, these microtubules form cilia and flagella. This formation is commonly referred as a 9+2 arrangement. Cilia and flagella help the cell to travel in the surrounding aqueous environment. Chromosome movements in cell division. In the cell division, microtubules pull sister chromatids of chromosomes towards the opposite poles of the cell to form nuclei. Organelle movement. Microfilaments. They are also known as actin filaments. Microfilaments are composed of linear polymers of G-actin proteins that intertwine into F-actin chains. Functions of microfilaments: maintenance of cell shape. They are tension-bearing elements. Changes in cell shape. The cytoskeleton can also contract, allowing cells to migrate. Muscle contraction. Here, myosin molecular motors collectively exert forces on parallel actin filaments. Muscle contraction starts from nerve impulses. We will talk more about muscle contraction in future videos. Cytoplasmic streaming. It is also known as cyclosis. It is mainly seen in larger cells such as plant cells. It is the active movement of constituents in a cell with the help of cytoskeleton. Here, myosin motors bind and push along actin filaments to move organelles along microfilaments. Cell motility, as in pseudopodia. Cell division in animal cells. In the stage of cytokinesis of mitosis, a cleavage furrow forms. This is made up of microfilaments. This divides the cell and produces 2 genetically identical daughter cells. Intermediate filaments. Fibrous proteins supercoiled into thicker cables contribute to form intermediate filaments. They are about 10 nanometers in diameter. These filaments are more stable than microfilaments. The type of the protein subunit that make up the intermediate filaments depend on the cell type. Some of the proteins are example keratin, nuclear lamines, vimentin, neurofilament proteins. Functions of intermediate filaments: maintaining cell shape, tension-bearing elements, anchorage of nucleus and certain other organelles, formation of nuclear lamina, participate in some cell-to-cell, cell-to-matrix junctions. Now, let's list out the major functions of cytoskeleton. Provide strength to the cytoplasm, encourage organelles and cytosolic enzymes of the cell, movement of cytoplasm, cytoplasmic streaming, positioning organelles, and move chromosomes when necessary, maintaining the shape of the cell, mainly in animal cells. Cilia. Cilia are short cellular projections that are often organized in rows. There are two types of cilia as motile and non-motile cilia. Non-motile cilia are also known as primary cilia. Most cells only possess a single primary cilium. Primary cilia function as a cellular antenna that coordinate many cellular signaling pathways and receiving sensory information. Motile cilia can be found in large numbers on the surface of the cell. They have a rhythmic waving or beating motion. They contribute to maintain the optimal levels of extracellular fluid bathing the cell. The structure of cilia is made up of microtubules. Here, 9 doublets of microtubules arranged in a ring with 2 single microtubules in its center. This arrangement is called 9+2 structure. Cilium is surrounded by a membrane contiguous with the plasma membrane but different in composition. At the base of the cilium, it bounds to a basal body which is a microtubule organizing center. Basal body anchors the cilium or flagellum to the cell. It has 9 sets of triplet microtubules arranged in a ring. This arrangement is called 9+0 arrangement because there are no microtubules in its center. 2 of the basal body's triplet microtubules extend to become the doublet microtubules of the cilium. Each doublet is connected to each other by the protein dynein. When ATP activates the motor domain of dynein, it attempts to walk along the adjoining microtubule doublet. This would force the adjacent doublets to slide over one another, resulting in the motion of cilia. Flagella. Flagella are long elongated structures that protrude from the cell body. They act as locomotor appendages that generate motion of a cellular level. 3 types of flagella have identified as bacterial, archaeal and eukaryotic flagella. Eukaryotic flagella have microtubules and dynein in the structure of their flagella, but bacterial and archaeal flagella do not have. In this section, we will talk about eukaryotic flagella. Cilia and flagella share a common structure, but cilia are more numerous than flagella on the cell surface. An eukaryotic flagellum has 9+2 arrangement, which is formed by a bundle of 9 pairs of microtubule doublets surrounding 2 central single microtubules. Flagellum is covered by the plasma membrane and is connected to the basal body, which has 9+0 arrangement as in cilia. The pairs of dynein arms in the doublet microtubules produce force to support the motion of flagella. This mechanism is same as in cilia. The energy needed for the motion of cilia and flagella is given through the ATP hydrolysis. So that's all today. Hope you enjoyed this video and gain lot of interesting facts about cytoskeleton, cilia and flagella. for your biological knowledge. So please subscribe this channel and click the bell icon. See you in the next video.